everyone. And welcome to today's webinar hosted by ReSViNET Foundation as part of the Clarity project. Thank you very much for joining us today. Clarity project is an EU-funded project with the mission to generate preventive and therapeutic approaches for chronic respiratory diseases. And the mission is knowing the molecular mechanisms, how viral infections change airway function, and how this will enable preventive treatments. And of course, the ultimate goal is to impact the understanding and also prevention and possibly treatment of virus-triggered asthma pathogenesis. Today, we will explore the genetic basis of virus infection-induced asthma, a topic that helps us better understand why some individuals experience more or severe or long-term respiratory outcomes following infections such as RSV infection. I am pleased to introduce our speakers for today, both from the Institute of Genomics at University of Tartu, Erik Abner and Grete Wil, who will guide us through the latest insights from large-scale genetic and biobank studies. Before we begin, just a quick reminder that you can submit your questions at any time the presentation, also afterwards, via the Q&A chat, and we will address them during the discussion at the end. And with that, let's start the webinar. Erik, please take over.
Sure thing. Hey. Can you see my slides and can you hear me well? And the pointer is visible as well.
Yes.
Excellent, thank you. So good evening and afternoon, depending on the time zone. My name is Erik Abner. I'm a research fellow of functional genomics in University of Tartu, so in Estonia. And exactly as was introduced, I'm going to be telling a little bit about how viral infections in general, or pathogens actually even, affect chronic lung traits, especially then focusing more on asthma. And as mentioned as well, it's also part of the project Clarity, where we're participating, and we're inputting then our side, from our side, the genomics information and the genomic analysis into this project. From my background as well, I did my PhD working on HIV, and so doing mostly lab work and trying to understand the epigenetics of it. However, now during my past 7-8 years, I've refocused into bioinformatics, and I'm analyzing now human genomics data. And I'm especially exactly interested in understanding the duality, understanding how the pathogens affect us and how they can lead us to have then chronic diseases, or how they have the long-term effect on our health. So moving on. So a little bit more about infectious diseases. It's, they're all around us. By the latest CDC, US Center of Disease Control statistics, there's around 1,400 pathogens afflicting people nowadays. Around 400 of them are the more common ones, and around 200 of them are viruses. But there's also, of course, different bacteria, protozoa, there's different parasites, fungi, whatnot. So there's a lot of stuff attacking us constantly, which means our body has also developed certain ways to fight against them. But it's this type of Red Queen hypothesis in nature that whenever there is a way that our body figures out to defend against the disease, the virus or the pathogens comes up with a new way to attack us. And so there's this endless back and forth happening between the different between the different hosts and the infectious traits as well. So it's a bit of like this tug of war. I like to think of it this way. And why I'm speaking of this is exactly because we have to understand it's a core concept of how pathogens function. They don't function in their own vacuum. They don't reproduce on their own. They need us to reproduce. We provide them with some type of sustenance that is necessary for their upkeep. Be it just for, maybe they steal some kind of nutrients from us, but yet more often than not, they actually steal some components from our body. You reuse them in their own way and then reproduce based on that, and then go find another host to infect. And so a lot of it boils down to genetic variation. And very often people, when we speak of genetic variation in context of infectious disease, they jump right into the idea of the viral or the pathogenic variation. As we saw, for example, in case of COVID, starting in 2020, if everyone remembers, there was the different strains. No, there was we started out with the alpha, beta, gamma, and delta strains, and later on they developed into Omicron strains, which we've been actually having for the last 5 years now. And then the Omicron strains in the past 5 years have diversified massively. And it's absolutely normal. This is how pathogens evolve. This is how they move forward. This is how they evade our immune surveillance as well. And that's how they keep on surviving from season to another season. However, there's the other side of the coin as well. And the idea is that humans also have their own genetic variation. It's not that, we're not all identical. Obviously, we are somehow different, all of us, which means that sometimes some viruses or pathogens, they will use that system as well against us. They will figure out ways how to find people who are more susceptible to their mechanisms, to their attack mechanisms, and then they will try to abuse those systems to then procreate on their own a lot more efficiently. And so that brings us then to the human genetics. So a little bit of background on that as well, because I'm guessing a lot of people didn't have their high school genetics recently. And so most basic level, summarizing it quickly, is that we're almost identical genetically speaking. If I take a random person, whoever is in this auditorium right now, and I compare that person to myself, we're going to be 99% identical. So we're extremely, extremely similar. And what I mean by similarity or this genetic variability is that if we just take this human genome, that's this 3.1 billion nucleotides, so those letters, and we compare them side by side, we see that those letters, this genetic code, were almost identical at every point, except for this 0.1% where we differ from each other a little bit. And these are the little differences that actually make us, those little 0.1%. But other than that, we're extremely similar. And so those little differences are, again, as we go back to a high school level, very often it's those simple traits. It's, it's the eye color that gets taught always. It's the height that is also extremely genetic. Nowadays, also, we're speaking, for example, of chronotypes. So what time do you wake up in the morning or what time do you go to bed? That can be very genetic as well. This natural cycle where you function or how you function. Another very, very well-known genetic system is how we perceive drugs, how drugs behave in our body. Not only drugs, but for example, how I digest coffee. What kind of kick do I get from the caffeine? But then again, not only that, it's also the genetics that starts playing into how our body fights against infections, or how it maintains itself, how it sustains itself against those diseases, and again, sometimes how those certain pathogens can actually abuse systems that we have actually, due to some natural variation. And also, what I would like to mention here as well is something that what gets taught in school very often is that genetics does very few things. I would like to highlight very early on that everything is genetic. At least that's my point of view, being in genomics maybe, but I cannot think of a single human trait that is not at least in some way governed by genetics. So there's going to be genetics everywhere. Everything we can think of, genetics plays a role there. It's just a question of how much does the genetics play a role in there. Is it 100% or is it just 5% of the trait that is genetic, inherently, coded into us? But for example, if we go to clinical genetics, like this most typical type of genetics when we think of disease genetics, is we see that we have those variants that are really rare. So if we look at this axis over here, so how common is a genetic variant and how high is the effect, like how strong is the disease. We see that usually clinical genetics focuses on this corner over here. So these are genetic variants that are usually extremely rare. We don't see them on average anywhere. Like these are not variants that tend to propagate within the population, and they do have this extremely high effect as well. So people with those variants tend to be sick, meaning we notice them a lot more easier. They end up in the clinic a lot more often. And then that's why they get certain level of attention. But these are not the variants that actually are the predominant variants in the whole population. So there's this whole lot of different types of groups of genetic variants. For example, the ones that are really, really common and don't really have much of an effect or have an intermediate effect. There's variants that are intermediate as well. So around 1% of the population has them, or those different levels. And these variants are the ones that science is very often focused on nowadays as well, trying to understand what they do and like how do they function. And maybe just to finish off this specific theme, let's going back to this high school type of genetics is it's a term called polygenicity, meaning how like one trait can be affected by multiple different genes. Again, high school genetics very often tells us, oh, there's this one gene, this one genetic variant that gives us the blue eyes and the brown eyes. However, nowadays, at least this simplistic model that is very often taught in schools, as mentioned, it's an oversimplification of the reality. So what happens in reality is we know that there's at least 20 different genes that will affect the eye color of a person. So you can have a person who has the blue eye gene, let's say, but maybe because there's this other 19 genetic variants, they can overpower that blue variant gene, and that person can still have dark eyes, or vice versa. The person can have the typical looking genetics for the dark eyes, yet have light eyes still, because again, it's a matter of combination between those 20 different genetic variants that we know of at least. And what happens very often is that these are exactly those modest type of genetic variants. So these are not the ones that we have that end up in the clinical genetics. It's not the ones in this cloud. It's the ones that are more common, that have a lower effect, but they are more numerous in the population. They do get their weight and their effect because of that. And modern science very often, especially the genetic science, then focuses on those variants and trying to understand how those variants affect each other and how they affect our functioning as well, this accumulation of those different variants all together. And so why do I mention this genetics and why I'm introducing it so heavily here as well? It's also part of it because we call the central dogma of biology, again, something from high school maybe, but the idea being that from genetics, from the genes, we produce the RNA. From the RNA, we produce proteins. Why is that important? Because the proteins then, well, they are essential. We can think of them as little molecular machines. And the idea is that they carry out all those little different processes that are necessary in our body. So they are necessary for, building different structures. They send the different signals, biochemical signals in our body. They move around different molecules. They recognize threats. They regulate the immune responses in our body as well. So they do a lot of different things that, makes us us. Of course, in the process, they also copy the DNA and they, replicate us. They create this system, allowing us then to live as we live. So it's important to understand this key concept here as well. Exactly, that this genetics leads to those proteins. Why does that matter? Is because proteins get hijacked by those pathogens that I mentioned earlier. So exactly. So those different proteins that our body uses, for example, for immune regulation. Maybe some viruses will use that. They will use that to enter the cells that we have, and then replicate inside there, and then carry out their own life cycles. So they will use those proteins specifically that we have. And here's where the genetics comes in as well, because if we have a genetic variant in a certain gene, that leads then to a different protein structure or some type of variation in the protein that we have. And then that will affect directly how the different pathogens get affected within our body, how they behave, what's their life cycle like. And so, for example, one of the most famous examples, maybe in infections biology and human genetics, is between HIV and a gene called CCR5. So CCR5 itself is an immune gene, doesn't really have a very well-known mechanisms. It's thought to protect us usually against different infectious diseases, somehow regulates how the immune system functions. What we know is that when that gene is dysfunctional, when it doesn't work, it doesn't really affect our health too much either. So it seems to be type of a gene that might have been crucial at some point in our evolution, but is not, it's almost redundant nowadays. We don't really need it. And that's what happens around 1% of Northern Europeans. So we know that around 1% of the Europeans up north, the more north you go, the more common it is. Those people are almost immune to HIV. How does that happen? So if there's HIV attacking our body, this is our cell, then CCR5 receptor is on the surface. HIV uses that receptor to enter into the cell. So it finds that very specific protein and uses that as a door essentially to enter the cell. And it can only do that through the CCR5 receptor, at least very specific, the most common strains of HIV in Europe. That's the receptor they use. And so among those 1% of Europeans, that gene is dysfunctional. It doesn't work. It doesn't produce a functional protein. That means that HIV actually loses the specific door it needs to enter, meaning the virus cannot really enter the cells, meaning people almost essentially are immune, at least to those common types of HIV strains. And so it's a good example where we know that this host genetics, this human genetics, affects how we get sick or who gets sick or how ill they become as well in the process, because again, we have those natural experiments almost where 1% of people don't get sick, and meaning they don't get the repercussions of getting sick as well from an infectious disease, at least. So it's essentially creating natural experiments for us in also for other traits that might follow this initial infection event. And so here we come in with our genetic analysis. So this is the title here is the GWAS study. So that means genome-wide association studies. So that is a very common tool used nowadays in field of genetics. And so what we do over there is we take large data sets. We take, for example, in this very specific case, that it's an analysis I did before, is we take around 22,000 people who have had Lyme disease in their lifetime. Lyme disease is a bacterial infection that you get after sometimes after getting bitten by ticks, so in the bushes, in the grass, and the trees. And we take a control group being people who never had Lyme disease in their medical records. And this is exactly, we don't have to recruit those people either. We can just use medical records that is almost publicly available to us, to the scientists. And then what we do as a part of a GWAS study is we just compare those two groups of people to each other, and we compare them based on their genetics. So we take these people have all been genotyped, so we know what their genetics looks like. It's from exactly all those 200,000 people. We have their genetic background, and we compare each and every genetic variant that we have from those people, and we just compare which genetic variant shows up more often, which one shows up less often. And just statistically running it through, and we'll see if any of the genetic variants show up really, really, really often or really rarely among those different groups. And then we can assume that, okay, if that variant shows up really, really often, that this variant is somehow causal, or it somehow affects the way this pathogen behaves in this organism. And so in case of Lyme disease, we did that. So here's something we call a Manhattan plot. Manhattan plot, it displays every little dot that you see here is a genetic variant. So it's one SNP. It's an allele. It's one little effect. It's one of those T, C, G, A changes. It's this minor effect. And so we did this GWAS study where we compared 30 million different genetic variants among Estonians against each other. So we just checked which genetic variants showed up more often. We found one very specific variant, and the higher the dots climb on this plot, we call it the peak. The higher that peak rises, the more confident we are that this variant does something significant in context of, in this case, in context of Lyme disease. And so we can then look in deeper, okay, trying to understand, okay, so this very specific variant, what does it do? We see that, for example, people who are heterozygous carriers of this variant, they have around 25% increased risk of getting Lyme disease in their lifetime. And the ones that are homozygous carriers, they have around 35% increase of getting Lyme disease in their lifetime. So it's an extremely significant effect, at least from statistics point of view. It's very clear cut. However, we do have to remind exactly it's this 35% increase. It's not a life or death situation per se, but it is still a remarkable risk factor. And that we have to remember in this case as well, that these variants, like the variant carriers for this specific variant, at least in Estonia, it's around 50% of the population. So half the people in Estonia, and pretty much half of people almost in the entire Europe actually, carry this variant, meaning they are extremely susceptible to getting Lyme disease when the tick bites them. So it's important to understand that exactly as well, that it's not just that this variant then displays a for example, we know that this variant doesn't affect people in any other way as well. We don't see those people having any other disease other than the fact that they get Lyme disease more often. So again, it's very contextual as well. So this variant, so we wouldn't even predict this variant to be disease causing in any way in those people as well. It's just in case of infectious diseases that we do see this effect. And this is the type of studies we do nowadays a lot as well. It's trying to combining these millions and millions of genetic variants we have from different population biobanks. We just do those heavy statistics number crunching, and we end up with some specific results. And from there, we can start interpreting what's happening and what types of like biological mechanisms are affected by those genes that we identify then. So it's like maybe what we can think of it as more like using genetics as a tool to understand how diseases are affected by pathogens, as opposed to the genetics being the outcome. Like we identify a certain group of people that are infected or immune or whatnot. It's more about understanding the biology behind the whole story, how this interaction between pathogen and the host happens. And so moving on to Project Clarity, which is where we have now been working for around 2.5 years almost, and what we're doing here. So as you can expect, we're focusing mostly on the host genetics. So we're trying to understand what aspects of human genome, what aspects of us affects then RSV susceptibility, RSV being respiratory syncytial virus. So we're trying to understand what are the genetic factors that RSV uses from us? What are important elements? What are the important biological mechanisms? And we are doing that in context of other chronic diseases as well, because there's been this long hypothesis in the air where it's thought that severe early life RSV makes people more susceptible to developing asthma or other related chronic lung diseases later in life. And so if, as I mentioned before, if all the traits that we can think of have a genetic component to them, here we can then do some nifty statistical analysis to figure out if that mechanism is truly causal, if RSV infections do truly cause those chronic respiratory phenotypes, or if they're genetics independent, or if it's completely separate themes altogether. And what we're doing then is we're doing a lot of GWAS studies to begin with. So we're GWASing every possible chronic respiratory trait we can think of. And we're also then doing a lot of analysis trying to figure out what is RSV interested in us as well. Like what are the genetic factors that predispose us to getting RSV more often, or what are the genetic factors that make us get the more severe effect, for example. And then understanding both the chronic disease genetics and understanding the RSV specific genetics, we try to overlap those results then, and trying to see which of those mechanisms then are shared between the two different trait groups, which are not. And that will help us then to decipher the causality mechanisms in there. And not only the causality per se, but it's also understanding what are the specific mechanisms there, what are the biological pathways, what are the individual players, because that's where we develop drugs from. That's where we develop this deeper understanding of the disease. That's how we can intervene if we have to in future. It's about understanding the story better. And so how do we do that? Biobanks is a word I mentioned already earlier. And so biobanks, they're, as written here, it's those types of large biorepositories where different samples and data is usually stored from volunteers. So here are listed 15 of the largest biobanks in the world that are more commonly referred to as well in at least in the scientific world. Estonia being your Estonian biobank, but I should definitely mention that the FinGen project is very notable. UK Biobank is very notable. And in US that we collaborate a lot with is a Million Veterans Program as well. And so altogether, if we combine those 4 biobanks that I mentioned, that gives us a sample size of over 1.2 million people, almost 1.3 million people, if I remember correctly. So we would have, just by combining different public databases and doing this type of statistics, we can then have the information from 1.3 million people in our statistical analysis. And again, we have the genetic data from those 1.3 million people as well available. So we can do the genetic analysis as well at the same time. And so what's the data that they do have? Usually it's medical records. That's the baseline that all those different biobanks would have. But then again, they also have very often some kind of drug prescription data. They have those behavioral questionnaire data. So what do people report themselves? Different laboratory measurements sometimes, cholesterols or some inflammatory markers from the blood. And so we can compare all that among those people. We don't have to only stick to asthma or some kind of very specific virus-specific trait. We can compare it all together and build this complex picture that then later helps us decipher the whole story. So here's an example that we have done. Here I already found a typo. There's a 1 missing. So, for example, it's a genetic analysis study, so the GWAS study that we did on asthma. So we compared 4 different biobank data sets altogether. With it, we have 156,000 cases of asthma, so different people with asthma in it. And we have 1.2 million people essentially who are the control group. And we compare the genetics of those 2 different populations. What do we get out of it? We get a very messy looking Manhattan plot. But then again, as I mentioned earlier, so each time those dots climb higher, that gives us the confidence that those genetic regions or those genetic variants, they play a very significant role in the disease. And over here, we can at least see 60 different genes or loci playing a role then in the development of asthma. But if we look deeper into the data, we can find even more than 60. Just not everything is drawn out on this very specific plot. And for those genetics and biology aficionados, what do we see here as well? We see a lot of immune phenotypes. So if anyone looks around those very specific genes, they will recognize interleukins, interleukin 33, interleukin 1 receptors. So these are very specific immune mechanisms. We see the HLA genes, TSLP, again, very, very specific immune regulations, as we would expect with asthma. But for example, I would also like to highlight the gene that is called GSDMB. It's gasdermin B. Why is gasdermin B interesting? It's because it's related to another type of immune mechanism where cells are told essentially by the immune systems to kill themselves. But it's not done, for fun or anything. It's done because when cells get infected, the immune system recognizes that this infection event has happened in another cell, and the immune system tells the cells to suicide essentially for the sake of the organism, because otherwise the pathogen within the cell can grow further and start infecting everyone else. So it's a defense mechanisms. And we see this GSDMB show up as well in asthma. Why I'm highlighting GSDMB is because we have another GWAS that this is based purely on the Finnish data set, so from the FinGen project in Finland, where we have 2,800 cases of bronchiolitis. Bronchiolitis is a very specific type of inflammation within the lungs. This lower, where the inflammation goes into the deeper parts of the lungs, the bronchioles. And so the idea is that we do a GWAS over here as well, and then we get a little cleaner GWAS, 1 gene. And it's the same GSDMB variant, so the gasdermin B gene that showed up in the asthma as well. So we already see early on that there's this very, very specific genetic link between asthma and bronchiolitis. So that gives us an idea that, first of all, there's a shared pathology from a genetics point of view between those 2 different traits. And now if we start adding here on top the RSV information, so we understand how RSV functions in the host, it gives us a very specific target to look for as well. And so what we are doing now, and it's very much ongoing work right now as well. I should mention we still have we're not even halfway through with the project. But what we are doing now is we are at least focusing on the Estonian biobank dataset that we have easy access to in Estonia, we now have done GWASs for over 500 different traits. We are going through different medical records, everything that we can relate to lung, respiratory traits, upper respiratory tract, lower respiratory tract, chronic, acute, infectious diseases, autoimmune mechanisms, anything that we can fix with lung functioning, we are analyzing from a genetic point of view, because all together it gives us the additional understanding exactly how those different traits function and what are the biological mechanisms governing that whole sector, the human body. So it helps us understand that. It's medical records combined together. It's drug prescriptions. So, for example, the inhalers that asthmatics are forced to use, procedures. So, for example, tonsillectomy is an important one, even though it's not upper respiratory tract per se. It's still it's not this lung-related trait. It is still upper respiratory tract, meaning it can still have any relevance. And it's an infection trait as well. Lab measurements. So we have those inflammatory markers. We have data on how often people get infected with different traits. So we're GWASing all that, combining all together. And so, for example, we have influenza already analyzed. We can see for certain genotype for people who get infected more often with influenza. We see the same thing for COVID, for example. A very specific genotype of people tend to get infected with COVID more often, or there's also another genotype where people that who get infected more severely. Also, another surprising thing we saw was that between the Omicron strain, that is the strain that is now passing around, and the earlier strains of COVID, those different types of strains of COVID, they were attacking different subsets of the human population. So there's a different specificity essentially for the different types of COVIDs as well. It essentially adapted to infect different type of people than the original, the alpha, beta, and the gamma versions were infecting. So there's this evolution built into it as well. And we can see that from the host genetics. So we can see that from the human genetic side. It's a fascinating world that we can analyze using all this data. Now, what is currently missing, of course, that I'm not showing here is the RSV data, because that's the layer that Estonian Biobank currently doesn't have. But Greta, my colleague, will tell you about it in some minutes. What are the other traits? So we're doing, as I mentioned, all respiratory traits. Anything we can think of that is somehow related to respiratory tract. Again, it gives us this background information, trying to understand what are the biological mechanisms governing those traits. And that's what we're doing right now, is compiling all this data together. And then, as I mentioned, we have those at least 15 biobanks abroad as well that are that also have this data available to researchers at least. We'll try to combine data from them as well with our data sets, with the idea that we can then create this massive, highly powered data sets that allow us to do a lot of different statistical analysis as well. Because a lot of those methods, they do require the statistical power that maybe one biobank will not be able to cover on its own. And so different analysis that we are going to be doing is, or that we are doing already, is genetic correlations. So we can genetically correlate one disease to another. So we can see if asthma and bronchiolitis look the same. If the people who have this high susceptibility to asthma and the people who have high susceptibility to bronchiolitis, are they genetically similar or not? Because that's then going to allow us to answer as well the question of the genetic susceptibility, especially if we can then bring in the RSV genetics to the picture. We can see if maybe they're both purely genetic traits, and there's the RSV doesn't matter too much in there. It's not. I'm not saying that it is that way. It's just that this is a question that could be answered with this types of analysis. We can just take individual genetic variants from those stories as well. We can, as I mentioned earlier with the CCR5, it's a very specific example, and the holy grail for such an analysis would be to find maybe there is a group of people who are immune to RSV or who are highly susceptible to RSV. In any case, it would be very, very good to know if there's groups of people that are differently affected in a strong manner by RSV. So that's something we're doing as well. There's more niftier analysis as well that are this bioinformatic approaches. So, for example, Mendelian randomization, where you can show causality between the different traits between each other, and using the genetic instruments from all the different analysis. Pathway analysis, again, that is important for understanding the way the different drugs affect us. What are the biological mechanisms within us that are being overtaken by the viruses or the different diseases? Because again, that's where the drugs come in. And of course, we know from previous studies as well what are the different genes and proteins that RSV could be attacking as well. So we try to then corroborate those information tidbits as well. And so take-home message just for my section, at least, is exactly that. Genetics in this very specific context, it's this project, it's more about understanding the biological mechanisms. It's about creating this background image of the human upper respiratory tract traits. And then we integrate that with the pathogen type of data as well, and that will give us a more complete picture. It gives us a better understanding on what RSV does to our body and why it does the things it does. And this feeds in later into the Project Clarity into other teams as well, because they are doing then the drug development, they are doing the more functional analysis in the laboratories. So those groups then can also verify the results we find, and again, that's where this understanding for this RSV to asthma causality comes in. So with that, I'm going to give a word to my colleague Grete.
Yes. Okay. I hope everybody can hear me. Hello, also from me. My name is Grete Lyll, and today I will give you an overview of how to use biobanks as tools to understand the biological mechanisms behind RSV virus. And my biology background is biology and gene technology, but I have been away from human genetics for quite some time. Since over the past few years, I have been mainly working on the human microbiome field, as you can see from the pictures behind me. But recently I have found my way back to human genetics, and I am very excited to be back. Now, Erik already gave you an overview of what the biobanks are. So today I would like to introduce you more specifically to Estonian Biobank, which is situated under the University of Tartu in Estonia. And as the name states, it is a population-based biobank that currently has more than 200,000 participants, which is roughly around 20% of whole Estonians' adult population. And Estonian Biobank, so to say, provides bread and butter to many, many researchers here who are unraveling the effects that DNA has on human health. Estonian Biobank was launched in 2001, but the idea for it came already in the previous century in 1999. And by 2004, Estonian Biobank already had 10,000 participants. And within the next 10 years, this number increased to 50,000. And in 2018, we had a second big recruitment phase. And during this, the number of participants increased to 200,000. And this year, actually, Estonian Biobank celebrated its 25th birthday. And during this time, a lot of different data sets have become part of the biobank. Many questionnaire data about diet, lifestyle, mental health, as well as many different omics levels. For example, whole genome sequencing, metabolites data, microbiome sequencing, and all this here is regulated by the Estonian Human Genes Research Act. And it's also very important to note that Estonia is a very digital country, which also means that our health system is heavily digital, meaning that all the prescriptions are done digitally, as well as we have countrywide digital health records, including procedures that the person has had done and which diagnoses they have had. And all this data is also accessible to researchers in Estonian Biobank. And we can work with this. And of course, all this data is pseudonymized. Now, before we look closer on how biobanks help us with unraveling the connections between infections and human genetics, we take a small step back, and I will do a little recap on what does the blood contain and what exactly are the antibodies that we are going to look at in this project. So let's start with blood plasma. Blood plasma is a liquid portion of the blood after it has been drawn and centrifuged, and it is mostly made out of water and dissolvent proteins, electrolytes, nutrients, different hormones, as well as waste products. And the plasma serves as a transport medium for blood cells and dissolved substances. It also helps to maintain blood pressure and supports immune function. And as I said, it also includes plasma proteins, including globulins. And antibodies are immunoglobulins. So they are these proteins that are being produced by immune system to recognize and neutralize different viruses, bacteria, and other foreign substances. And each antibody is highly specific and binds only to the pathogen it was created to fight. And do note, in Estonian Biobank, we have plasma samples that were collected between 2004 and 2024. And the idea here is to use antibody detection to determine whether an individual has had contact with RSV. And if these RSV-specific antibodies are present, it indicates that the immune system indeed has had a previous contact with the virus. On this slide is a figure from which you can see how this process of RSV infection, antibody response, and antibody detection looks like. So first, when the RSV attacks body, it enters our organism through nose or mouth, and it invades the epithelial cells, which leads to infection, which is then a trigger to immune system to tell that we are being under attack. We have to do something, which then activates B cells that begin to multiply and differentiate into plasma cells, as well as start producing antibodies that are specific to RSV antigens. And these antibodies that are being made are going to start circulating in our bloodstream. And so when the blood plasma is being collected, if these antibodies are already circulating in the body, they are also going to be inside the blood sample that is being collected. And this can be then determined with ELISA kit. So how ELISA, which is enzyme linked immunoabsorbent assay, works is that they have this wells, and this bottom of the well is coated with RSV specific antigen. And now when this sample is being added, if there is an RSV specific antibody present, this will bind itself to this existing antigens. If they're not present in the sample, then nothing binds to these antigens that are being coated into the well. And after that, this another enzyme conjugated secondary antibody is added to the mix, and this then binds to the antigen, if there was any. And what is the purpose of this secondary enzyme conjugated antibody is that it converts into a colored product. So in case the color appears, it means that yes, this sample had RSV specific antibodies in them, and this person has had a recent or ongoing infection of RSV. However, if we find no color appears, then this states us that this person to whom the sample belongs to, they don't have RSV infection in their body. And when you start thinking like that, okay, but what about this RSV vaccines? They also vaccines work the way that they're gonna, introduce antibodies to your body, specifically IgG, and it's in our Estonian Biobank, it's highly unlikely that we're going to get some response due to vaccinations, because RSV vaccines became available in the market in 2023. So all anti-RSV antibodies that we are going to be able to maybe detect in our sample set, they are probably only from natural infection exposure. I'm sorry, the colors here are not how they were before, but if we think that we want to select individuals who might be infected with RSV, then we also have to take into account that RSV circulation is very seasonal. So here is a plot from June to June again, and it shows that the RSV levels begin to increase in late October, and that they usually peak around December or February. And IgG antibodies peak then usually 2 to 4 weeks after symptoms start and the exposure to RSV virus. So here on the bottom, we can see one example that if an RSV exposure occurs, then it takes about 7 to 10 days after the exposure for IgG antibody to become detectable. And as said, it peaks after 2 to 4 weeks, and by week 8, it is still detectable, but the titer begins to decline, and the decline continues over the next months. And now when we're coming back to biobanks and how to find RSV infected people from here, as I said, we have more than 200,000 samples that we can use to detect these RSV specific antibodies. And based on the previous slide, we will concentrate on the samples that were collected during the infectious seasons from December to May. May is added in because there we added a little like a buffer zone to the antibody development. So in total in our biobank, there are more than 85,000 samples that are collected during these months. And they're not just from year 2018, they're over the course of 20 years, so from different years. And now our current aim is from these 85,000 people to choose around 20,000 samples or 20,000 individuals for RSV specific antibody testing. And our expected outcome is to link lower or higher antibody levels to host genetic variants. So we want to see if a person who has a very high level of RSV antibodies compared to the low, if they have some differences between their genetics. And as said in the beginning, these regular integrations of biobank samples with electronic health records enables retrospective tracking of different diseases, including respiratory events. So when choosing these 20,000 individuals for RSV antibody testing with this ELISA kit, we will be mining the electronic health records for symptom clusters consistent with RSV infections, such as fever, cough, rhinorrhea, sinusitis. And here our idea is also to exclude the individuals who have, during the time they gave their sample, diagnosis of influenza, swine flu, or COVID-19. And this is done to reduce the confounding factor. And if you ask, like, why are we looking for RSV diagnosis, then because we only have adults, and adults very rarely make it to the doctors with RSV virus because usually the symptoms aren't that severe. Their symptoms of RSV are severe in children, but not in adults. So this is why we have to go and make a very big data mining to find these people to choose to this RSV antibody testing, because we don't have like a clear diagnosis for this here, because people usually don't make it to the doctor with this. And our plan is also to add in asthma patients into this antibody testing. Around 28,000 individuals in Estonian biobank have asthma diagnosis. And adding in approximately 1/3 of the samples originate from these patients. And we also want to add in some other respiratory or lung disease patients to have comparison between the respiratory disease cases as well as unaffected individuals. And with the asthma patients, one idea is also to include inhaler purchases as a potential proxy for how severe this asthma is, because if we can see that one person has a lot of purchases for inhalers, this might mean that they have a more severe asthma. And here I brought out the hit for bronchiolitis that Erik already showed before. So previous GWASs, as said before by Erik, have identified multiple genetic loci associated with susceptibility to asthma or bronchiolitis, implicating genes involved in immune regulation, airway inflammation, and antiviral host responses. And given the established role that RSV infection in early life bronchiolitis and its proposed contribution to later asthma development, we also sought to investigate whether individuals exhibiting this high specific RSV-specific antibody titers, if they also show genetic variations within these previously found asthma and bronchiolitis-associated loci. So we want to see if this, yes, I said this high RSV-specific antibody people, if we compare their genetic to this with low ones, if they're gonna come up with the same loci that have been found previously. And it's also very important to note that RSV is also genetically complex as any disease. And it is probably also an interplay between multiple genes that are just like one single genetic determinant.
Later,
and
can I shortly just interrupt? So we have 5 minutes left. Maybe if you can, yeah, this is I think this is the last slide. Is that correct?
Yes, this is the last slide
okay.
for me. And my take-home message is actually they're pretty much exactly the same that Erik had. So we can skip it because it's exactly the same, just like different wording.
Yeah. Okay. Thank you very much, Grete, and also thank you very much to our both speakers. Actually, indeed, submit your questions in Q&A. I have one question, and it says actually, how did you define high versus low RSV antibody levels? Have you developed any ways to specify what levels of antibody are associated with recent infection and what recent means? Is that 1 year or 3 months? And did you use a commercial or an in-house RSV antibody assay?
So
I guess I can take that.
Yeah,
sorry.
Go ahead.
Sorry. Yeah, so I guess I can take that. I'll start from the end, actually. So commercial or in-house RSV. We are going to go with a commercial one. So as Grete mentioned, the idea is to do around 10 to 20,000 blood samples. And why this number is, well, one thing is budget-wise. Second thing, commercial is more reliable. So it does give us a certain reproducibility as well, if necessary. And also at the same time, we're not going to go in blind. We're now testing different types of kits to see which one actually works best with the samples we have in the biobank. And regarding the recent infection and the infection level, so that's another data layer that we have in the biobank. So we do know the IgG and the antibody levels in general of the biobank participants from different time points. We can compare the different IgG levels that the person usually would have. So if they are, highly producing or lowly producing, And we can also then time it. So when the sample was collected and how far away it is from, for example, the infectious season, or usually the virus peaks in December, January, February, a little bit of March as well, I guess, is the end tail. So we can try to then see as well when the sample is collected after the last viral season. And so it's and it's a numbers game in that sense that there's a chance that a lot of people are not going to be infected with RSV. So there's going to be just a very low residual effect from the IgG pattern, so that they maybe were not infected within that season. They were infected maybe in the previous season before that. So hence the reason for having a lot of a large sample size, because that then allows you to decipher between the the inaccuracies that are going to come from this type of sampling.
So thank you for that. I think this is a major database, and really great to see how we can use this for getting closer to the answers to our questions. There are a few other questions, but unfortunately we don't have time at this. The last slides are now gone, but I would like to thank everybody who joined, and also Eric and Grete. Thank you very much for your presentation, and also this very beautiful data. And in the meantime, I would like to ask everyone who attended to fill out the questionnaire. There is a very short questionnaire after the webinar. That would be very helpful for us to improve. And there will also be a replay available on ReSViNET website in the coming week. So then you can look back and also share with others who couldn't join for today.